need to have all of these combining elements, and they're way smaller. So how fast can you replicate a prokaryotic?
Really fast.
Really fast. Does anybody know how fast typically like staph replicates? It take days, weeks, hours, minutes? Minutes. For them to replicate their DNA, we're talking a couple of minutes. Okay, staph on average generation time is about 35 minutes. Okay, so this is explaining why. It is so few nucleotides, it's going to run very quickly. And it doesn't have any of those same problems that linear chromosomes will have. How do we feel now about this?
Much better.
So, okay, I just want to clarify with the last one. So each human chromosome is longer, and they have many origins, because if they just had one, it would take forever to replicate that.
Perfect.
Okay.
And human chromosomes or any animal chromosomes they're linear. They're not all the same size. So some have thousands of genes on them, some have hundreds. In men, there's one that has like 6. The Y chromosome has like the SRY gene, and that's it, basically.
So for number 4, it doesn't need the lagging strand, right? Just to clarify.
Correct, because it does need it's just going to build, just goes.
Okay, thank you.
So it doesn't need primase or DNA polymerase 1. All right, is there any questions on this worksheet that anybody would like to go over? We'll start on page 1. Stop me if anybody has a question.
Can we go over 6?
6. Okay. Can it start at any exact moment?
I can start where the bubbles are, right?
Okay. So let's do our annotation, all right? So we have initiation bubble right here. Therefore, there has to be a sequence right there. We have another bubble right there, so it has to be right there. Here we have a bubble, so there has to be one right there. And then we have another one right here. So how many points of initiation do we have on this chromosome?
4.
Excellent. And so all of them will have those. What happened at this guy? This was another quicker question we had.
They combined.
Yeah.
Bind.
The bubbles work together.
That will happen too. What enzyme causes them to do that?
Ligase.
Caleb's like, I got ligase. We know this. Okay, how we feel?
Feel okay.
Okay. We'll keep going down. Any more questions on this page?
So for 6, is it the DNA synthesis starts with specific locations then?
Correct.
Is it? Okay.
Specific locations, which are called
Your initiation?
Initiation point. Points of initiation, yes. And what binds to them?
Initiator protein.
Excellent.
Question, is the initiation point the same as the origin of replication?
Yes.
Okay.
Okay.
I hadn't checked this, but I heard people in the Discord talking about how the link doesn't work or they're having issues with it.
The link, yeah, there's a space weird written out here. If you just Google this thing, it pops right up.
You have
Yeah.
to put molecular dash visualizations dash of dash DNA.
Okay, or they changed the damn link.
Mm-hmm.
You know how it is. No one can keep anything the same. Best part, I think I updated that link last year on there, so. Okay, how I feel? Ready to move on?
Yes.
I'm
Okay,
still a little bit confused on number 3 on that page. I mean, 4.
four, got it. Who is this?
Valentina.
Okay, sorry. Just trying to keep track of who I'm talking to. So we have a new type of enzyme, very similar to DNA polymerase 3, but it does not need a primer to start because it can just start anywhere. So this is very similar to what other enzyme do we have that can do that? Give you a hint, transcription.
RNA polymerase?
Excellent. RNA polymerase does not need a primer to start. It just goes. So this would be very similar to RNA polymerase. So if I don't need a primer to start, I can just start anywhere and build me a leading strand. So therefore, I do not need DNA polymerase 1 because I'm not replacing any primers. And I don't need primase because I don't have any primers. Does that help?
So is he asking which one we don't need, or?
So it is asking which ones you do need. So you need ligase
Okay.
and helicase, right?
Okay.
I'm just saying which ones you don't need. So
Okay.
you need a helicase because do you still need to unzip DNA?
Yeah.
Yes, right? And you would still need ligase to merge any of those replication strands.
And then you don't need the polymerase or the primase because you don't need to. You can start anywhere.
Correct.
And for the diagram, we wouldn't need to draw any lagging strands because they just go away.
Basically, because it can start anywhere and unzip and go.
Okay.
All right. Get rid of my notes so you guys just can't see the answer when it pops up on the screen. All right. Question for you guys. Couple more seconds. Okay. On what side do you always build on?
3 prime?
3 prime. There you go. And we'll go to the next question. So make sure to respawn the answer. Okay, closing up. There's still a couple people who haven't answered. Going. All right. Okay, a couple more seconds. All right. What end am I building on?
3 prime?
We're not building on a 3 prime end. That's the problem, right? So
We're
I need,
building on a 5 prime end.
we're building on a 5 prime end, so can I do that? No. So I need to put down a primer at the end so then I can run it backwards. So I can build on a 3 prime end. So I need primase to put down something at the very end. Then I need polymerase to rebuild it and from 3 to the 5 prime end. And then I need ligase to put them together. So I need all 3 of those. Okay. How we feel?
Can you repeat that again?
So which side of this strand am I building on right here? Can you see
The
my
prime.
arrow?
Prime.
Yeah, that's the 5 prime. So I have to build on a 5 prime end. I cannot do that. So what I'm going to do is can you guys see my arrow? Just asking.
Yes.
My cursor, okay. So I'm going to put down a primer right there. So now I have created a 3 prime end, so I can use polymerase to run backwards. Then I'm going to ligate it right there. The problem is, let's say the primer's 5 nucleotides long. Can I replace those 5 nucleotides? No, that's our end replication problem. So that's where we need telomerase to be able to do it, to extend it out. Otherwise, chromosomes get shorter and shorter and shorter through time. All right, so for this answer, all 3 of them.
So it was primase to be able to add something to the 5 prime, and then the polymerase is to be able to replace it, or to.
Sort of. We're not adding to the 5 prime end, because that all just creates a new 5 prime end. We're going to put one down here at the other end.
Okay.
This Okazaki fragment we're creating. Right, so I'm going to put one down here. Then it's going to elongate. We're going to run it back into it, so it's a new fork, right? And then we're going to ligate it so that the phosphodiester bonds get created. Okay?
Question. So, do you say it's telomerase that adds strands?
It adds on to the end of a strand, correct?
Okay, and then DNA polymerase, it builds within that where the five carbon is.
DNA polymerase is going to run from the primer to the 5 prime end, and then that 5 prime is going to connect with the 3 prime, and we're going to use ligase to build them together.
Gotcha.
Okie dokie. Are we good to move on, or we need more time?
Yes, we're good.
Anybody else? This is the problem with Zoom. I can't see your faces, so I don't know how lost we really are in the real world.
I want to double check. So polymerase, what was it for? Is it to?
Polymerase builds DNA.
Okay.
So we're going to put down a primer so I can create a 3 prime end.
Yes.
From there, polymerase is going to build off that 3 prime end as long as it possibly can. But it's going to run into that 5 prime end pretty quickly. And so when it hits that 5 prime end, it's done. It falls off. And then ligase will come in and connect those 2 bonds.
Okay, thank you.
I want to make sure we're doing good and understand this.
You said earlier that this is how old age happens, is because they
Yeah.
slowly get shorter over time.
Yeah.
But
Yeah.
it looks almost as if by what you're saying, then they don't get shorter, or is this not a thing that regularly happens within DNA?
Not a regular thing. It's only in some cells. Typically, stem cells. Stem cells and gamete cells are pretty much the only thing because they start as stem cells. So your skin should not be able to build telomerase, should not be able to stop this problem. So it should age. Aging is fine because cells will just eventually die. The problem is when they activate all these things and they're able to elongate, those cells will do uncontrolled cell division. Okay, so every once in a while when we're putting down these DNA strands, a mutation occurs. When they occur, we call that what?
Can you show them?
New allele.
Caleb just wanted to say a damn new allele. I think that was Carrie. That we. What'd you say?
I asked if you could show the meme on the previous slide. Thanks.
Yeah, you're welcome. Mutations create a new allele. Every time it happens, it's a new allele. Does not matter. It's a new allele. All right. Sometimes it happens and it does nothing. Right. It's still a new allele, but it did not change anything. Right. We have redundancies in the codon, hence the meme. I was going to get to that. So if we look at the meme here, change in the base pair did not change the codon sequence. Okay. So if we went ACC to ACA, that might be a different codon, might not be. And so they may or may not change the phenotype. If they changed an amino acid, it could be a change in the phenotype. So on the exam, for instance, we had valine, we normally had glutamate, GLU, was substituted for valine. And what happened is you had a hydrophilic R group, now becomes hydrophobic. That's going to change our outcome, just like we did in lab, right? So we had cystic fibrosis, sickle cell, hemochromatosis, all of those change the phenotypes. You could have it change the whole damn protein sequence, right? Mutations are extremely rare, okay? I'm going to give you a lie. The lie is mutations are random. Every year there's about 4 or 5 lies through the class that we have to tell you. This is one of those lies we told you. Mutations are random. Turns out they're not random, okay? But for all extensive purposes, the enzymes that are putting things down, DNA polymerase 1 or 3, does not make mistakes very often. When they do, it's a mutation. It should be random. They're not always random. So there's certain chemical processes and molecular techniques that actually cause mutations to increase or decrease and the likelihood. But when they do, it's really hard because let's imagine you have an adenine and it replaces on the opposite side with a guanine, right? Not only do there's supposed to be 2 bonds versus 3, they're not in the right orientation. They're literally in the wrong stereochemistry. So what happens is DNA is going to there's DNA polymerase is going to run along the backbone, and anytime it detects a weird shape. So if I do this, now the backbone is a slightly different shape, and it's going to find it because the backbone is literally bulging out. And so when it finds the bulge, it just cuts it out and replaces it. So when a mutation does happen, it gets replaced very quickly. But every once in a while, you can get a mutation that fits in the right patterning. So mispairs, for instance, if we look, T to G can happen. If we're going to get a mutation, it's going to be T to G. Okay, that's just the likelihood. And if we look at their bonding and how it can work, that's why, because we don't have any covalently locked carbons preventing it. C to A can happen. And that is just because we're not, the stereochemistry doesn't prevent it. So, in summary, mutations are extremely rare. They create new alleles. There is redundancy in the code, so mutations should be reduced down their effect, right? So it might be in a codon, might not. The other thing is there's tons of DNA that actually doesn't transcribe anything. We're going to talk about that here in a second, what that is. We call it junk DNA. There's no such thing as junk DNA. But we're going to call it junk DNA for a second. But when it does happen, we can predict pretty much what it's going to be. It's going to be a T to a G or a C to an A, just based off of stereochemistry and the fact that there's not a covalently bound carbon preventing it. How do we feel about that?
So I have a quick question, Ricky. Is the fact that the code is redundant, is the purpose of that evolutionarily because it helps reduce deleterious mutations, or no?
That's a great, fabulous question. I have no answer for you.
Cool.
We, as humans, really like to think of things as designed. We love this idea, concept of design. Probably it's just random luck.
It just seems funny that it's so redundant, but I don't know enough about it to know if it really seems funny or not, you know?
But the funny thing is not everything is redundant. So if you were to build like we build redundancy into a lot of stuff. We build redundancy in your cars, airplanes, so forth. But when we build redundancy, we do it for every system, every link in the chain, right? So I've heard this exact argument before for intelligent design. And my argument against this as redundant is if that was the case, the intelligence who designed this was lazy. Because if it was intelligently designed, it should have had redundancy in every system and every backbone, but it doesn't. So it could be intelligently designed and just the person be lazy. And there is actually a hypothesis of this, that we are all a simulation and it's a 12-year-old and we're ants, right? Like they're playing their video game. I'm going to go with no. You know,
I've heard that one.
For selfish purposes, I'm going to go with no. There are some very famous people who totally buy into this simulation idea. But if it was, it's lazy. Yeah.
Thanks.
And the chemistry just works really well. So, yeah, I'm so sorry. I cut somebody off.
You're fine. I have a question. So the code's redundant. So if you were to change like the last letter in a codon, it wouldn't change anything. But what if you were to change?
No. That's only the case in some codons.
Okay.
Do we have a codon here? 1 second. Give me a second. Let's pull up a codon table so we can all see what we're doing.
I think one example is like AAA and AAG, which are both lysine. And if you just change out the last A for G, you still get lysine.
Right. So what happens when you change the first two?
Yeah, so give me a second. Can you guys see the code? There we go. There are certain ones where if you change the last one, you get changes as well. Okay, so alanine to met, phenylene to leucine. Leucine, if you look at LEU, has all kinds of them that correlate to leucine. You can change the first letter or the second or the third, right? Cysteine to arginine, cysteine, right, to tryptamine to stop, right? UGU to UGA is different. UGG is different. So that's where I would say the redundancy is lazy, because if you wanted redundancy, what you are saying would be correct, right? There would be more redundancy. There's not. There's redundancy in some of them, like alanine, for instance, tons of redundancy. But the other ones, not so much.
So I guess my question is, does this relate to like silent mutations, deleterious, things like that?
Yes, so a silent mutation does not show up in the phenotype. Right, so this goes to a question in your lab this week. If you have a mutation that is silent, is it recessive or dominant?
Recessive.
Recessive, because it does not show up in the
Phenotype.
phenotype. Okay.
Okay.
How are we doing for everybody else?
I had a question. What did you say specifically about the DNA polymerase 1 and 3?
DNA polymerase 1 replaces the primers, the RNA. DNA polymerase 3 builds the leading strand.
Okay, thank you.
So I have another question. When you were talking about when mutation happens, it usually gets replaced rather quickly. But sometimes it lands in a right sequence and it doesn't get detected.
Yep. That's when it will show up.
That's when it will show up, like.
That's when it will show up in the next cell. Like if it doesn't get replaced, now it can show up. Now it can be part of the phenotype. But not everything is a part of the phenotype. That's the redundancies.
Okay.
Yeah.
Let's see. Thank you.
Okay. Can we move on? All right, cool. We have a lot of genes. Okay, there are a ton of genes. Are all the genes in every single cell?
I'm thinking yes, but I don't know.
Yes, absolutely. All genes are present in every single cell. So the question becomes, why does a muscle cell have muscle stuff? And why does a neuron have neuron stuff?
Is it because certain genes are activated?
Beautiful. Certain genes are activated in certain cells. You have to have triggers to activate things. So, each type of cells. Yep.
In the reading that had like a certain name for like the trigger, like I guess what triggers like something to be active versus what triggers something to not be active. What were they called again?
So
I don't know if you know.
So there are different cells. We'll go through like totipotent, pluripotent, differentiated, determined, right? We're going to go through all those and what things turn on and which. And so then when a cell is determined or differentiated, it can activate by regulatory sequences, which we're going to talk about. Okay? So all genes are present in all cells, except there are exclusions. Red blood cells are an example where they get rid of everything. Red blood cells do not have any genes. They don't need it. They just get rid of stuff. So you want to make a cell, and the best way to describe this is a supermarket. So imagine the biggest damn grocery store you've ever been to in your life. Okay, that represents all of the each product is a gene. And I want you to make me a cake. How do you go get the ingredients to make a cake?
Look down
What do you
the
do when you
aisles.
get to the store? Okay, what are you looking for?
These certain ingredients?
Yeah, but what do you look for on the aisle?
Flour, sugar. Is that what you're asking?
Yeah, but what are you looking for? Like, what tells you that aisle has it?
The number on the aisle?
Excellent. Number on the aisle. The signage. The bigger the store, the more signs you need, right? So imagine I take that store and I flip it upside down, big giant monster, and I shake it a bunch. Will you ever be able to make me a cake?
Eventually, but it would take a long time.
Yeah, it would take you your whole lifetime, right, to find those products. This is gene regulation. Gene regulation is you need signage to tell you where to build things and when to build them. Okay, so let's imagine I'm walking through the supermarket, okay? And if you've probably experienced this in your lifetime, you get familiar with things. And it pisses you off when they decide to move something around because, you know, especially in your old age like me, I get grumpy and I'm like, where the hell's the flower? It used to be right there. But that's because we're used to signage. We're used to things put together. So for instance, if we want muscle tissue, we're probably going to put all the muscle stuff on the same aisle. And an aisle will represent a chromosome. And we will have that aisle available. Because if we want to build muscle tissue, we want that aisle available. So everything else can be put away. We can store it. We don't need it yet or need it now. So what we'll do to any other aisle that we don't need, we'll histonate it. We'll condense it down. We'll put it away. We'll store it off to the side. And the aisle that's needed is open. And then I'm going to put signage on it. I'm going to make the signs, because I'm dumb, okay? When I'm in a store, I'm really dumb. I need big signs with big arrows that tell me where to go, and I need lots of them. And so we're going to put signs everywhere. And those signs are going to be very specific sequences, just like we do at the grocery store. Those specific sequences are going to say, go here, go here, go here, here's your flower. And we're going to call those regulatory components. So in your genes, when we talked about it last quarter, there were structural genes and regulatory genes. Regulatory tells things when and how to build. Structural is how to do it. So the cake, the structural, is the flower. The regulatory is the sign hanging above the aisle saying flower. The tag on the shelf that says flower. The barcode on the gene that screams dummy, this is flower, right? Those are the regulatory components of a cell. So years ago, when they first started doing genomics, they discovered that there's like 50,000 genes, okay? But there's like enough DNA for 500,000 genes. And somebody's just like, oh, that's junk. It's historical junk. It's not needed. And some of that might be true. Some genes, like if you have a tail, we all have tail genes. Maybe they're junk. Maybe we don't need those anymore. They've been isolated off. We don't use them anymore. That could be true. That could be junk. But the rest of it, the vast majority of that junk is signage. It is the regulatory components saying where things are at. And then there's other ones that says how to build it. So I need not only to build my directions to build my cake, the ingredients. I need to be able to build it. And so that's all the other regulatory components. And so that's what we're going to look at for, we're going to start here today. I don't think we want to, being on Zoom, it's really hard to like, where do we want to get to? I was going to create some videos sometime today, tomorrow, to do the lac operon. And then we can give you guys some time to do the worksheet on your own, and then we'll discuss it in hopefully in person or on Friday or something. If that sounds fair to you guys, because how many of you like going into breakout rooms to do worksheets? Does anyone want to do that? Okay, so I think my idea might be a good idea. So we're going to think about regulatory components. And the one we're going to think about, and I'm trying to figure out how to do lab. We might be able to swing something where we can do it online. I'm going to think about it tonight and try to figure something out. Is called the lac operon. And we have a worksheet, and I did not send it out yet. We wanted the worksheet we just finished. The lac operon, we're going to start with a prokaryotic gene regulation worksheet. And then next week, we're going to dive into eukaryotic gene regulation. Prokaryotic gene regulation is a single circular chromosome. So imagine instead of this giant big supermarket, which is the eukaryotic, the prokaryotic is like a convenience store, like a really tiny little gas station convenience store. One aisle, okay? Not even multiple aisles, one aisle. So if you needed all the ingredients, could you find them really easily in that one aisle? So how much signage do you think you need?
Not much.
not much signage. Okay, and so okay, no, I want annotate. Oh, because I'm sharing. I'm sorry, guys. I'm not very good at Zoom. Used to be better at it, not anymore. I threatened after COVID. I was like, I will quit. I'm never doing this again. We have our single chromosome. Okay. So if you were just to say, okay, I need a gene. I need gene flower. Okay. Gene flower happens to be right there. But you don't know that from afar. So you're going to walk around that chromosome. So you need to find it. How you're going to find it is a little flag that says, this is gene flower. This is called a promoter. The promoter is a sequence that tells it to build. We already did this. Okay? So that sequence is on your DNA.
So, the promoter can literally be like the marketing where it promotes it.
Correct.
Okay.
You have your -35, -10, +1. This is your gene. This is called your promoter. Okay? That's it. On the simplest level, that is it. It tells it, build here, we're done. The problem is, do you want to build everything?
No.
I give you the ingredient flour. How many things can you build with flour?
A lot.
A lot. And as somebody who likes food, that's pretty awesome. The problem is how full is your kitchen going to be? Really full, right? So we don't want that. Very, very full. Like, this is going to go badly. So what we're going to do is we're going to make it very specific. Okay? We're only going to turn things on and off when we need them. So if I need flour, yay, let's use flour. If I don't need flour, we're going to
store it.
Not use flour. We're going to store it away. And so there's ways to store things permanently, long term. That's called histonated. In eukaryotics, we histonate it. We wrap it up and we throw it away. We histonate it and we methylate it. We'll talk about that later. For those that have kind of, dabbled into more of biology and science in their life, this also can be called epigenetics. We'll talk about that later. But it is strong or weak. Okay? So if this promoter, if we want to make a gene all the time, and let's say it's a really, really important gene that we're going to make it all the time, we're going to make it a strong gene. Okay? We're going to make the promoter bind to the RNA polymerase often. Okay? We're going to make it often. But if you need it, that needs to be made occasionally, you're going to make the promoter weak. So it'll only bind occasionally. But then there are genes that you don't want on very often. And those are ones we're going to regulate with positive and negative regulation. So let's imagine what we're going to do in lab is called the lac operon. The operon is just a barcoded gene, is what we call it. And we call it an operon because this is controlled by an operator. Right here we have CAP. We'll talk about that in a second. Then we have the operator. No, sorry, the promoter. And then we have the operator. And then we have a gene. You have something called a repressor and an activator. A repressor stops it. It acts as a brake. The promoter or the gas pedal will accelerate things. It'll make it go faster or slower. So imagine this is a car. Okay, this is actually called the car model. you are trying to drive your car. You have a brake and you have a gas pedal. If the brake is on, will the car ever move forward? No.
No.
Nope.
So that's called a repressor. So if the repressor is on, car never moves forward. RNA polymerase is the car. The car wants to move forward, but can't. The brake is on. But if the repressor is off, can the car move forward?
Yeah.
Yeah, can it move forward at different speeds?
Yep.
Well, if you have your car, let's say you're driving your car, you take the brake off, but you don't push down on the gas, what happens?
You just keep it kind of like neutral.
It goes because it's still in gear.
Stand.
It just goes. It's just going. It's just slowly going, right? But if I throw the gas pedal down, now it goes
Fast.
But if I have the gas pedal and the brake on at the same time, what happens?
Burnout.
Nothing. You don't move. You just spin those tires.
Donuts.
Possibly. Which is a fabulous time of year right now. Just saying, I might have done some this morning.
Good.
I had to take Sylvie into town because she watches being watched by my mom. And on the way, there was an empty parking lot. And I was like, yeah.
Yeah. Did she like it?
No, she wasn't in the car. Just me and Boz. And he did not like it.
Oh.
Yeah. So this is gene regulation. That's all it is. So I have a repressor and we have an activator. The activator is called cyclic AMP, CAMP. CAMP buys the CAMP. That makes it go faster. That's your gas pedal. The repressor is a protein that binds to the operator. It tells it not to go. And that is your brake. Okay, so let's imagine you have a gene that makes something, I don't know. Let's say makes brown sugar. And you are trying to bake an omelet. Do we need to have the brown sugar?
I hope not.
I hope not. So what is going? Are we turning the gene on or are we turning the gene off?
Turning it off.
Turning it off. So we have the repressor on. We are not going to use this gene. So the brake is on. But let's say you've done with your omelet and you want some, I don't know, what do we use brown sugar? I'm going down a rabbit hole here. We're going to make cookies, okay? We want the brown sugar. So I'm going to remove the repressor. We're going to take the brake off. Okay? How do you feel about that?
Good.
Okay. In both of these cases, we're going to make something. So in the case of the omelet and the cookies, we're both going to need a frying pan. So we need to have a frying pan. Do I need lots? No, I'm going to need the oven, okay? Just the stove. We're going to make the stove. We're going to make this work. In this case, I want the stove being made, but do I need lots of stoves?
No.
No. So I'm not going to have a repressor. We're not going to have an operator. We're just going to make this slowly. So we're going to make it the sequence strong or weak?
Weak.
Weak.
A weak
Excellent.
sequence.
A weak sequence. All right. I have a question for you guys. In this just for a second so I can load something.
Is this in a new session?
It is. All right, just a couple more seconds. All right. So, what do we think it means to be weak?
It doesn't get expressed all the time.
Okay, excellent. And what does that mean? We still want it to be made all the time, but we want to make it slowly all the time.
There's less promoters.
It's not less promoter, but the promoter itself is weak, so it's going to be a different sequence. So let's imagine a strong promoter is exactly what you guys have seen before, okay? It is called your ATA and TATA boxes, okay? So your promoter is TA, T, T, A, so forth, okay? So then the opposite would be that, okay? That's your -10. This is called your promoter. And you have an enzyme that knows how to bind to this very well, okay? It can bind to that. That's RNA, that's going to bind your RNA polymerase. Okay, so this protein is going to bind to it. Let's imagine, and this makes, a lot. The new sequence, and this is what we call a strong sequence, it binds to it. The new sequence is this. So the opposite is this. Will will that sequence be recognizable?
No.
Well, sort of. Is it very similar?
Yeah.
It's pretty similar, right? But it's a little different.
Except the fourth nucleotide is
less often. That's going to be a weak. So this would be strong. This would be called weak. It can bind to it. This certain shape, right, will bind, but not as good. So just like your enzymes did, it'll catalyze the reaction slower. That's called weak. So the promoter has a specific sequence, and if it does not match, it's called weak. How do we feel about that?
Good.
Okay. So let's apply this. So guys, re-answer. Let's apply it to a new principle. Going. All right.
Just to confirm, what was the correct answer on that one?
A specific sequence. So if it's not the correct sequence, it will be weak.
Okay.
So I believe it was 4. What do you think is going to have a strong promoter?
right.
So the question says which one's going to have a strong promoter? A tRNA gene, which makes what, guys? What does a tRNA gene make?
Polypeptide chains.
It makes tRNAs, right? You need tRNAs to make your polypeptides. Does every cell in the body need to make polypeptides?
Yes.
Yes. Every cell in the body needs tRNAs. Does every cell in the body need an enzyme to convert intermediate amino acids into glycolysis?
No.
No.
So the one that's going to be made all the time is tRNAs. Something that's going to be made less often will be weaker. So tRNA gene is going to be very strong. I have a question. So let's think about it. This would be a great exam question. Hint, hint, hint. Which gene in a cell do you think has the strongest promoter? Which gene in a cell do you think has the strongest promoter?
AUG?
No, because AUG is a codon. It's not making a
Wouldn't
gene.
it be like mRNA or something?
What do you mean mRNA? Because mRNA is what we're making on everything, right? That's the point here. That's transcription. But
Okay.
I want to make something. What do I need to make? That's the most important thing to make.
Ribosomes?
Ribosomes. Beautiful. Because don't you need ribosomes to make everything?
Yes.
Yes.
So that is going to have the strongest of all promoters. Is the gene that makes ribosomes. Okie dokie. Does anyone have any questions?
So ribosomes are just made up of a bunch of genes? Maybe that's a really dumb question.
A gene makes ribosomes. Genes make ribosomes. And you need to be able to transcribe a gene to make a ribosome. So does the gene that makes a ribosome have a promoter?
Absolutely.
Yes.
Absolutely. Is it going to be strong or weak?
Strong.
Really strong, because we need to make that ribosome all the time. You have a gene that makes eye pigment. Okay? So you have a gene that makes eye pigment. Anything that gets made on your body has a gene. Okay? So I just picked one that makes eye pigment. Is that going to have a strong or a weak promoter?
A strong promoter?
Eye pigment?
A weak one?
Weak, because how many of your cells need to make eye pigment?
Not all of them.
Very few.
Not all of them.
1%.
So, yeah, not even 1%, like 1/1000000000 of 1/1000000000 percent, right? Like a few cells in your eyes need to make eye pigment. Literally just your cornea. So it'll be weak. And it will have a lot of regulatory components to it. Because do I want to make that pigment?
Not very often.
No,
Or
not
no.
very often. So we're going to shut that shit down. But if I'm making ribosomes, I want to make them all the time. We're going to turn those babies on and they're going to have a strong promoter. So what I'd like you to try to attempt to do by Friday is the prokaryotic gene regulation worksheet is on Canvas. And then Friday in our office hours, that's what we're going to go over. Okay?
I have a question about the car model. We were talking about the repressor and then the activator, and then how like if you're not pressing on the gas, you're still moving. Is that just basically like the reaction being spontaneous?
It is not spontaneous. So what happens in your car if you take the brake off?
Well, I don't know. It depends on like the transmission, because
Okay, transmission's engaged, okay? We're trying to go forward. What happens to your car?
it still roll.
It goes forward, right?
Yeah.
It's going forward slowly. That's what's happening. So we don't have the gas on. We're not making tons of it. We're not going fast. It's just the brake is off. So it can move forward. It's going to move forward very slowly.
So is that like with the example of making cookies or whatever, you're just kind of letting it roll, you're not, because you don't want it, you're doing it slowly because it's not necessary all the time?
Yeah, so it's something you need, but you don't need a lot of it,
Okay.
right? But if you need a lot of it, you put it down on the gas.
So like for making ribosomes, you'd have the gas on, and then for making the eye pigment, you just let it slowly.
No, you probably have the brake on.
Oh, okay.
Because do you need to make eye pigment?
No.
No. Only
Would it
like
be-
10 cells in your whole body needs to make the eye pigment. And you only made that eye pigment how often in your life?
Probably once.
Once, when you were a baby, right? And so if you didn't need it, you don't need it now. So we're good. Shut that thing off. Put the
But
brake on.
okay, but it was on while it was making, and then it shut off, or
Correct. The brake came off for a moment in
Okay.
time when it was making it. And then
Okay.
once we made it, we don't need it anymore, so we shut it off.
So is it kind of like going to the grocery store just that one time, rather than like making a whole bunch of trips and
Yeah.
going there over and over because you need to keep doing it?
So what's something that you'd only buy once in, like, in a blue moon at the grocery store, right? So
lemon curd.
Like
What'd you say?
Seasoning.
Seasoning, cool. You bought it once. You made it once. Okay, like, you know, I'm trying to think of something even stupider than that, right? Like, yeah, we get it. We understand.
Lemon curd, I said.
There we go. And then once we don't need it anymore, we shut it down. We put a brake on it. We set it off. To
Who
the side.
shuts it down?
Itself.
The
It's
repressor?
self-regulating, right? The repressor does.
So what does it mean when an eye pigmentation darkens over time when you age? Is it just cells dying?
There's a couple of things. One is that it can be being made over time. It's the repressor never bound to it. It's the accumulation of pigment.
Is that like a slow process, like kind of
It
how you
is.
were saying that it's like that weak promoter, so it's happening slowly?
Correct.
Kids who like start out with like blue or grayish eyes and then they go to brown.
Right. So all babies come out with gray eyes for the most part. Over time, their pigment gets made. So that it is a very weak promoter, so it's being made slowly, but the repressor's off. At some point in your life, usually around 5-6 months, the repressor gets put on. That gene gets shut off for the rest of their life. And whatever eye color they have at 6 months, that's it. Every once in a while, a human, because we have variation, right, will accumulate eye pigment in their lifetime. What went wrong?
The repressor
There we
wasn't
go.
very strong.
The
Excellent.
repressor didn't hold on to it.
Correct. The repressor did not bind.
Need to change the brakes or what?
Yeah, they have no brakes. Yeah, their brake line got cut.
What's the
same-
difference
so things can
between
happen. Okay, so we're gonna go down a rant. Okay. In biology, there are 3 sexes: male, female, and intersex. Okay, that is just a fundamental fact of life. Intersex has a variation of things. Okay, intersex can mean everything from slight androgenism to true hermaphroditism. Often, this is a mistake in a promoter or a regulator. So there are populations of humans that are born female. They are female until about age 12, because that repressor from the SRY gene on the male chromosome never turned on. So the repressor never came off. The first hormonal spurge that they got from their adrenal cortex that released a little bit of, the pituitary released a little bit of hormones, testosterone. That SRY gene now got activated. And they will grow a penis. And they'll be about 12 years old. So that is a great example of gene regulation. There are many different types within that, within animals. So in humans, there's like 60 different variations of intersex because of different regulatory components. So I got another one. You are XY. Biologically, you are XY. The problem is your whole life, you were phenotypically female. What never turned on?
Y gene.
The Y.
Y
Y
gene.
chromosome?
We never made that single protein. We make that single protein. All of a sudden, that single protein turns on. Guess what happens? Your phenotype completely changes. We have other examples. So you could be XXY, right? You could be any number of different things. And it comes down to regulation. It's all about gene regulation. So our hormones trigger the repressors or trigger the activators. Things get lost. They get turned off. So there we go. That rabbit hole. But it's a really good example.
So for like the sex changes type of things where you can take certain medication that changes your hormones, would that be it like turning the gas on?
It could be if you have the genes to activate. So trans is different than intersex. You can be trans and, you know, behaviorally, psychologically, you feel like you are a male, but you're biologically a woman. Okay, that is independent of what I'm talking about. Mm-hmm. Those people, if you give hormones to, their tissue might respond as if it was a male, but they don't have the SRY gene to make the structures like a penis.
Okay.
If they are born female, but are biologically male, meaning they have XY, you could trigger that gene by activating it. Absolutely.
Is there a way to identify that, like, without activating it to
do a karyotype map? You can
Oh.
literally look at somebody's chromosomes to see what they have.
Okay.
Yeah.
You said that hormones are the triggers for those promoters for determining sex and all of that.
For a lot of times, yes.
What are other triggers for the promoters?
So, in this context or just in general?
I guess kind of in general, like what would that look like?
Let's talk about the lac operon. The lac operon, this is what you're going to do in your worksheet. Okay, and this is what we're going to do in lab. We have our gas, our promoter, and we have our operator, so that's our brake. And then we have 3 genes, X, Y, and whatever.
Kenneth?
And we'll call it X, Y, Z. I can't remember what they are. It might be X, Y, Z. The brake is on all the time. Okay? When do I want to make the lactase enzyme?
When there's no glucose?
It's not when there's no glucose. It's when there is
lactose.
Lactose. Excellent. So if we lactose present, so let's imagine that is lactose, okay? So we have a beta-galactosidase linkage right there. If that is present, it's going to bind to the receptor, causing the receptor to fall off. Or sorry, the repressor to fall off. So in this case, the activator of the gene is lactose being present. Because lactose, when it's there, removes the repressor. It allosterically binds to the repressor, causing it to change shape, which causes it to fall off. So any number of things can cause this. So for instance, there are certain people in that have no ability to eat maltose, very similar, because their repressor is bound to the DNA and doesn't fall off in the presence of maltose or ethanol. So any number of things can trigger a repressor or change it. So an example would be lactose, maltose, whatever, fructose, as well as other triggers, hormones, testosterone, estrogen, estradiol, cholesterols, all of those can trigger things to change. Okay, how do you feel?
So when lactose is present, the repressor falls off and it goes.
Correct.
There's no break.
There's no break.
Okay.
So could it go fast or could it go slow?
It's gonna go fast.
It can go either, right? All it means is
That depends on the promoter.
settle down yet.
Oh, okay.
So you have positive regulation and you have negative regulation. The break is negative regulation. Positive regulation is the gas is on and the brake is off. Then that thing is just going to move really quick.
You were talking about the car model. You mentioned, you said something binds to cap or something. What
Yes,
about
CAMP. CAMP binds the cap. You'll learn all about it in the worksheet.
So cap binds to, what was it?
Cap.
And then binds to what?
So the cap binding site, cyclic AMP binds the cap.
Okay.
Yeah, and that helps recruit by sigma, which binds to the promoter, to the cap binding region. So in the readings, so the pre-lab was the LAC-OP run. Please do that, even if we don't have lab tomorrow. So we're not having lab tomorrow, just, I don't see it happening, especially because the weather, it's just gonna get worse. So what I think we're gonna do is, I'm going to probably do a recording with data, and then we'll probably do or we might just do lab next week. I haven't figured this out yet. We'll figure it out.
When you say the worksheet, do you mean worksheet of the lab or a worksheet?
No,
of the
worksheet in class. So we have a worksheet.
For
it's
today?
called the lab. It's in Canvas, yes.
And that's for today, like what we should have done.
Yeah, so get that done. Try to do it by Friday. And I'd like to go over it Friday and Monday. So we'll do a little bit like in office hours. You guys come, we talk about the worksheet, and then on Monday we hammer this.
Let me also understand, for Friday, it's not in person. It's
on Zoom, like
it's on
normal.
Zoom. Okay.
I suspect, looking at the weather, just kind of what I'm hearing and like the things that they're sending out at the school. Tonight is supposed to be really awful for weather, and it's supposed to get pretty bad tomorrow as well, like just really cold, really icy. So I suspect nothing tomorrow, probably nothing Friday, but like by next week, hopefully Monday, it's okay. If not, we'll do this again on Monday.
I have a quick question before we end.
Yeah.
So in the readings, it's talking about like LAC-Z and LAC-Y and LAC-I. What exactly are those?
Those are the genes that are going to be made. So the LAC-I is the repressor. The LAC-Z and LAC-X and all that are the subunits of the lactase enzyme. Okay. So I make 3 mRNAs. How many polypeptides do I make?
1.
3.
Oh, 3.
How many subunits do I have?
3.
3.
3 subunits.
I have 3 genes here. How many subunits am I going to make?
3 subunits.
There we go.
And that'll be lac X, Y, and Z.
Yep.
Okay.
And that will make your enzyme called beta-galactosidase.
When you say a subunit, in my mind it comes the ribosome, but what's a subunit in this case? Or
Subunits is, so go back to the protein and enzyme worksheet. Subunits are the different chains, the different polypeptides. So we have quaternary structure. So 3 subunits, 3 N-termini, 3 C-termini, we are quaternary structure. If you have 2 subunits, you have 2 N-termini, 2 C-termini, you still have quaternary structure. If you only have 1 unit, you have 1 N-termini, 1 C-termini, therefore do you have any quaternary structure? Nope. So this is why we started with proteins, the biochemistry of proteins. We need to understand what a protein is, to understand how to build an enzyme. And now we're using enzymes to build
Genes.
the genes that to make the proteins, to make the enzymes. This is a circle.
Okay, so lac Z, Y, A
Yeah,
are transcribed into 1 mRNA as co-transcription.
Yep. The only pleasant thing about Zoom is I get to see all the animals. That's it.
This is what she does when I do homework. She lays and sits with me the whole time.
Excellent.
Mine too. She can't.
What's the cat's name?
Her name's Franklin.
Hi, Franklin.
Frankie for short.
Still good kitty.
She lays on my arm though when I'm like trying to type, and then she gets upset when I move.
Yeah.
It's a cycle.
She says, "You're my human."
Mhm. All the animals. Cool. All right. So, I guess to end us off, attempt a worksheet. We will debrief and make sure that we understand it really well. We'll figure out what we're doing for lab probably next week. So I will see you, no matter what, I will just see you on Friday on Zoom.
Sounds good.
Sounds good.
Thank you all for being so flexible today and coming up with nice ideas. I do appreciate it. It's kind of, a mess.
And thank you for your ideas as well.
So, cool.
Everybody have a snowball fight now.
Yes.
or don't make donuts though.
Go make donuts. There
No,
we go.
no donuts.
All the donuts.
We gotta make
No donuts.
Oh. No side.
But if you do, invite me.
Yeah.
Bye.
All right. I'm on if anybody has any questions.
I'll wait for a minute.
Bye-bye. Have a good day.
Bye-bye, guys.
I have a question.
I will, yeah. We have your exams graded, by the way.
Oh.
I forgot to say something.
Are you gonna post
I was getting
it?
in the back today.
Are you gonna post it or not? The grades.
Yeah, I'll send out the grades. Why not? What's your guys' questions? Nothing?
I was just wondering, for the essay reading, was it just one time